Hello everyone, I'm Jan Witkowski. I work here at Cold Spring Harbor Laboratory. I welcome you to the 86th Cold Spring Harbor Symposium on Quantitative Biology. And the topic this year is genome stability and integrity. And I'm very pleased to have Johannes from Harvard here. It's such a pleasure having people around after a gap of 2 years in this classic meeting.
Indeed. Couldn't agree more.
So you've not spoken yet, Johannes?
Correct.
Okay, so you can give us some background on your talk. And reading your abstract, it's clear that the key thing are these DNA protein cross-links, DPCs.
Mm-hmm.
How are these formed, and
right?
And what proteins are involved?
Uh-huh. Yeah, so there are a number of sources of DPCs. One source is highly reactive molecules that exist in cells. Probably the classic one would be a formaldehyde molecule. So every time that you demethylate a histone, you generate formaldehyde close to the DNA. And if you're unlucky, it can end up cross-linking a protein, a DNA binding protein, to the DNA that it's bound to. So that would be one source. But then, of course, there are highly reactive bifunctional compounds that are used in cancer therapy, such as cisplatin, that generate not only DNA interstrand crosslinks, where the two strands of the DNA are cross-linked to each other, but also they can cross-link proteins to DNA. And then sometimes proteins that act on DNA, like topoisomerases, that have a covalent intermediate as part of their enzymatic cycle, they can, if they get stuck and can't finish the cycle, then that can also lead to a DNA protein cross-link.
So how often does a topoisomerase do that? Is it
it's not clear. I think most of the time when we study trapped topo complexes, we induce them with a drug, such as camptothecin, for example. But clearly it does happen occasionally, as part of a normal enzymatic cycle.
And the proteins are always DNA binding proteins. They don't sort of grab some protein that's arbitrarily floating around close
I think
to the
in
DNA.
the vast majority of cases. So people have done mass spectrometry on the DNA protein crosslinks that are generated as a result of formaldehyde exposure, and the vast majority are DNA binding proteins, and actually most of them are histones, not surprisingly.
And the consequence of this is that does it interfere with separation of the DNA strands? Does it interfere with some enzyme coming along the strand? What are the functional consequences of these things?
Right. So the primary consequence is, of course, that it causes problems for the process of DNA replication. And when we started the work, we assumed that a large protein adduct on the same strand that the replicative helicase translocates along would permanently arrest that helicase. Okay? But then actually in the course of studying the collisions of replisomes with DPCs, we were very surprised to see that although the DPC transiently stalls the CMG helicase, it doesn't actually block it. And the helicase can in fact get past the DPC, and only after the helicase goes past does the real process of repair, that is the proteolysis of the protein adduct, actually initiate.
So these DPCs only get excised or dealt with in the course of replication. The cell doesn't have a scanning mechanism that surveys a DNA strand for?
That is an excellent question, and in fact, there's another group, Niels Meiland's group, in Copenhagen, who has uncovered what appears to be a replication-independent mechanism of DPC repair. It involves sumylation, so a sumo modification is added to the protein. And this then leads to a cascade of events that eventually removes the DPC. And I think you've hit on the key question, which is in a replication-independent pathway, what is the sensing mechanism? And I don't want to speak for him, but I think whenever I see him, I ask him this very question, and he they're working on it. One possibility would be transcription, but even if they block transcription, I believe this pathway still operates. So it's a fascinating question, what it is that senses these DPCs outside of the context of replication. But that's not what we work on. We ask what happens when a replication machine collides with a DPC.
And so the helicase, CMG helicase, is the first step of essentially of replication that's separating the strands.
That's correct. It's the machine that travels out front, right? And the polymerases follow behind and then copy the separated strands. So it's really the first element of the replosome that will collide with an adduct such as a DPC.
So what I'm trying to get my head around is the idea that you've got a double-stranded DNA that's got this protein glued to it.
Correct.
Helicase travels along as it separates the DNA strand goes through the helicase.
That's correct, right.
So how does it, the helicase is like this with the DNA strand, that's this great glob. How does it get, how does it get round it?
So the honest answer is we don't know. So what we do know is that the DPC that we're using is far too large to pass through the relatively small central channel. And so we speculate, and we're trying to test this, but we're not there yet. We speculate that the MCM ring, which forms the core of the CMG helicase, we hypothesize that it cracks open at one of the MCM interfaces, and that allows the helicase to pass the protein adduct through the gap. That's our current hypothesis for how that works.
So remind me, the helicase is made up of a set of subunits that form this ring.
That's correct.
So you feel that simply, well, I can say simply that that ring splits.
Correct.
Enough to allow the
right. That's the idea.
The block through, and then
that's right. And in fact, right. So it's formed of a ring of 6 related, so-called MCM ATPases, and so it's therefore very possible, I think even likely, that it's always the same interface, let's say between 4 and 7 or 2 and 6, that would open up when it hits an obstacle like this.
Do you think that's I was going to say deliberate design. One shouldn't say about deliberate design with molecules, but that is a feature of the helicase design that
has evolved in order to
cope with these things.
Yes, absolutely. And what's very, very interesting is that we already know of instances where the helicase cracks open, right? So when the MCMs are first loaded onto DNA in the G1 phase of the cell cycle, they're actually cracked open so that the double-stranded DNA can pass into the channel, right? And then you have 2 MCMs that are loaded head-to-head, and they encircle double-stranded DNA. But then when MCM becomes activated as a CMG helicase, now you actually have to extrude one of the 2 strands, because the active form of the helicase is in association with just 1 strand, right? So after you've loaded the MCM onto 2 strands, then in S phase, the helicase has to open up again, allow one to exit, so that now you can adopt the active conformation. And I will be very, very surprised if the ring cracking that allows you to get past an adduct will not occur at precisely the same interface as what leads to initiation in the first place.
Is that it's known where that thing, even in initiation?
Well, so it's known that the MCM2-5 interface opens to get the MCMs onto the double-stranded DNA, but when MCM is then converted into CMG, the MCM2-5 so-called gate is covered by two MCM cofactors called CDC45 and GINs that are required for helicase activity.
Right.
And so I think most people accept the idea that that gate is now permanently closed, and that the gate that would open for initiation and possibly for DPC bypass would be another gate that is actually not obstructed by CDC45 and GINs.
What's known about the strength of the interactions between these MCM subunits? I mean, have atomic,
Yes, so there are high-resolution structures, and it's actually known that before the MCM loads, that the 2-5 gate is particularly weak, and actually there's a gap. But I don't think that there's a very clear candidate for the others. But of course, the opening could be an active process, and in fact, John Diffley's group has shown very nicely that during the process of initiation, to get that strand out, you need another initiation factor called MCM10. So what MCM10 probably does is it destabilizes the interface between two of the other MCM subunits.
And you said the DPC that you use in your system is particularly large. Is it large in comparison with what one would expect to occur naturally? Is it abnormally large, I suppose?
I wouldn't say so. I mean, we actually use a bacterial DNA methyltransferase just for convenience, because it's very easy to covalently couple it to a specific site on DNA. And it's about 45 kilodaltons. So that is larger than a histone, obviously, but it's smaller than some other proteins. So I think it's kind of in the ballpark of what replisomes would often encounter.
Right. So now we've got the helicase past this roadblock.
Yes.
What triggers the proteolysis step?
Right. So until recently, we thought we had the answer, and we had part of the answer, which is that, so if we back up for a second, when the replosome hits a DPC, the CMG helicase is initially stuck right at the DPC before it goes past.
Right.
And the helicase is large, and it has a large footprint. And what it does is it imposes on the leading strand a fairly distal arrest point. So the leading strand can't get more than about 30 nucleotides closer to the DPC. So it's stuck at what we call the -30 position, okay? And once CMG goes past, then the leading strand can extend towards the DPC and stall 1 or 2 nucleotides from the DPC. And we've shown that extension of the leading strand to the DPC is critical to activate a protease called Spartan. And we think that the way that works is Spartan actually, it has a protease domain, but it also has a couple of DNA binding domains, one of which interacts with the protease domain. And we think that once the leading strand extends to the DPC, now this creates a structure that the DNA binding domains can bind to and de-repress the protease domain, okay? So that's where things stood for a couple of years. And then we started investigating another DNA helicase that we knew from mass spectrometry experiments also gets recruited to replisomes when they stall at DPCs. And this helicase is called FANCJ because it's mutated in a human genetic disease called Fanconi anemia. And we can talk about that later. And we thought, okay, well, since it's there, we might as well ask what it's doing. And I didn't mention this yet, but all of the experiments we do are in frog egg extracts, right?
Yes.
And so we use an antibody to get rid of the FANCJ protein out of the extract. And shockingly, we found that when we remove FANCJ, the Spartan protease is completely inactive. So it doesn't degrade the DPC. And long story short, we now think that after CMG bypasses the intact DPC and the leading strand gets extended to the DPC, we think that FANCJ loads onto the single-stranded DNA that now exists downstream of the DPC and translocates back towards the DPC, and using its ATPase activity and perhaps other clever tricks that we don't yet understand, it literally unfolds the DPC, okay? And that unfolding, we now think, is crucial also for Spartan activation.
These systems seem extraordinarily complicated. Although I suppose the old argument about design is that if you were going to design a system, this isn't what you would design. You would design something that was a bit more logical and rational.
Well, you say that, but and when we went into this project, right, we thought for sure the Helicase is going to hit the DPC, and once we knew there was a proteolysis event involved, we thought surely the DPC will be degraded to a short peptide that allows now the Helicase to go past, right? That made perfect sense. And then we discovered actually the Helicase goes past, and only then do you degrade the DPC, and that seemed very strange and paradoxical. But then, after thinking about it, we realized that this actually is highly advantageous because it means that the proteases will not inadvertently degrade the Helicase. Okay, so the Helicase is free and clear when you finally activate the proteases. And that's actually crucial because one thing we've learned over the last 20 years is that the way that cells limit replication to a single round per cell cycle is that once you're in S phase, you're not allowed to load any new helicases from scratch, right? You have to start the process in G1, and when you're in S phase, you can activate the origins that were licensed in G1, but you cannot recruit any new MCMs de novo. So if you should accidentally degrade your Helicase, that replication fork will be permanently stalled, and that could lead to a genomic catastrophe.
So are there DPCs that are meant to be there as part of, say, gene regulation or something, and that the cell that shouldn't be removed?
Are there covalent DPCs that exist as part of normal cellular metabolism that shouldn't be removed? Well, so the obvious examples, again, are the transient intermediates during topo isomerase-mediated relief of topological stress. But of course, that's an intermediate, and upon completion, right, of the enzymatic cycle, the DPC leaves. I can't actually now think of any DPC that would form and never leave the DNA. So there's actually a fascinating new protein called Helicase, which Dave Cortez characterized recently, which covalently attaches itself to a basic sites that exist in the DNA to protect them from further processing into double-stranded DNA breaks. And we just published a paper actually showing that after Helicase cross-links itself and protects the DNA, it's ultimately removed by Spartan using exactly the same mechanism that I just described.
Well, we must finish, but I did actually make a note to the effect that I noticed you use Xenopus extracts for your work, which I was about to say, what a throwback! I don't mean that. I don't mean that unkind. But of all the classic systems that have been used since the 70s for studying cell cycle activation and stuff, so they're still useful.
They are very useful. They are actually still the only system that can recapitulate the full cycle of vertebrate DNA replication in a test tube. And the wonderful thing about them is that they contain the entire frog egg proteome, and so it's really like a cell in a test tube, right? And in fact, it's a wonderful discovery tool, right? When we first put a DNA template with a DPC into these extracts and replicated them, we had no idea that any of this existed, right? It was only by doing this experiment in the egg extract system that we even discovered the concept of replication-coupled proteolysis. If you tried to do this with a reconstituted system, you would never discover it because you would never think to add all of these components. Right.
Oh, well, good for Xenopus. Johannes,
good for us.
Yes, well, true. Thank you very much.
It's my pleasure.